Alright, so here we are in the second to last chapter, and we'll be talking about enzymes and vitamins here in chapter 20 as we near the end of our course. Now, in this first lecture, we'll be focusing on enzymes and enzyme action. Obviously, the vitamins will come later on. Okay, so as you can see, our title of the slide says enzymes are biological catalysts, but we're a little glib in that first definitional sentence where we say enzymes are proteins, because not all enzymes are proteins. It's been known for about 30 years now that RNA can act as an enzyme, the so-called ribozyme activity there, and so there's our non-protein catalyst, biocatalysts, so non-protein enzymes that won the Nobel Prize back in 1989. So just be careful. We tend to focus on the proteins because they tend to be the most important and overwhelmingly majority type of enzymes, but just be aware that not all enzymes are indeed proteins. However, all enzymes do catalyze the chemical reactions taking place in the cells of the body, and they increase the rate of reaction by lowering the energy of activation, right? We can't really increase the rate by increasing temperature. There's a threshold where that doesn't work so well in the body. Once you get above 40 degrees Celsius, as we've mentioned before, the body doesn't tolerate that. The human body, anyway, does not tolerate temperature extremes beyond that. So instead, in order to make these reactions happen at a reasonable time frame so that we don't build up poisons in our cells that ultimately kill us, we have these catalysts, these enzymes, to allow the reactions to take place at a very reasonable rate and still at a normal body temperature. So how are these enzymes able to accomplish this amazing feat of lowering that activation energy and thereby increasing the rates of reactions? Well, nearly all enzymes are globular proteins, as I mentioned in the previous slide. There are even non-protein enzymes, and there are certainly non-globular protein enzymes, but the overwhelming majority of enzymes are protein in structure, and they are also particularly globular proteins in terms of classification. So within these enzyme tertiary structures, we have the active sites, the place where the small group or groups of reacting molecules known as substrates bind in order to allow that chemical reaction to happen in a very favorable manner. The whole purpose of that enzyme is to hold that substrate so that it's poised for reaction. That allows the reaction to happen at that decreased activation energy, which we mentioned allows the reaction to happen at a reasonable rate while still maintaining that ideal human body temperature. Okay, so if you recall the active site from that previous slide, that whole globular protein there is designed to hold that specific substrate. So it's many amino acids long, and most of that is just to get the folding right so that that active site is correct for that particular substrate. So enzymes are highly specific, and the specificity of these enzymes is sometimes absolute. So that means they can catalyze only one reaction for one specific substrate. Sometimes you can get a little generalized activity. I use that term loosely because you might be able to catalyze a reaction of two or more substrates, but not a whole bunch. And then some enzymes catalyze a reaction for a specific type of bond. So that's called a linkage type of enzyme specificity. So if we look at our table there at the bottom of the slide, table 20.1 shows that absolute specificity, which means just one type of reaction for one type of substrate, and we see urease catalyzes only the hydrolysis of urea, one specific substrate that it can act on for that particular reaction. For group specificity, we have the ability to catalyze one type of reaction for a group of similar substrates. Hexokinase adds a phosphate group to hexoses, so it's not specifically glucose. We have other hexoses that it can work on as well, so that's why it has a group specificity. And then finally, our linkage specific enzymes catalyze one type of reaction for a specific type of bond. We have chymotrypsin there that catalyzes the hydrolysis of peptide bonds, so it breaks down polypeptides or proteins by hydrolyzing that peptide or amide linkage, and it can do that to a a whole host of substrates because we have many different types of substrates that have amide linkages. Okay, so regardless of the type of enzyme specificity, whenever we have an enzyme catalyzed reaction, we get a combination of enzyme and substrate forming an enzyme substrate complex, and we denote that as a capital E, capital S. So that enzyme substrate complex provides an alternate pathway for the reaction with lower activation energy, so we don't have to increase temperature as we ordinarily would in chemistry in the lab setting, where the glassware doesn't mind going up to 100 or 200 degrees Celsius. The human body does mind that, so we instead allow these enzymes to catalyze reactions by offering these alternate pathways. So within that active site, we have the amino acid side chains. We mentioned that it's tertiary structure for these mostly globular proteins. They catalyze that reaction by having those side chains hold the substrate in just the right way to yield an enzyme product complex. So there we see the enzyme plus the substrate come together, and notice it's a reversible reaction to form the enzyme substrate complex. Then in a completion type reaction, that goes on to form the enzyme product complex, and finally in another completion type reaction we get the enzyme separate from the product. That enzyme is therefore free to go back and bind to another substrate and do the whole thing over again. That's what we mean by catalysis or a catalyst. It can act on a substrate and then act on a new substrate after it's done its work on that original substrate. So if we turn our attention to a particular type of enzyme substrate complex, let's look at enzyme substrate complexes for disaccharide hydrolysis. So here hydrolysis is accomplished by the enzyme sucrase, a molecule of sucrose. The naming is pretty easy here, right? Not like that beginning organic chemistry naming. Biochemistry naming tends to be pretty logical. So sucrase is the enzyme that acts on sucrose. So the molecule of sucrose binds to the active site of sucrase. That glycosidic bond of sucrose is in a favorable position for hydrolysis now because it's being held in just the right way by that enzyme. Therefore, it has a much lower activation barrier than if we were just allowing that sucrose molecule to bounce around freely in solution. And now we see that we have the sucrase plus sucrose. They're free in solution in an equilibrium type reversible reaction fashion. They form that enzyme substrate complex, the sucrase-sucrose complex here, enzyme named first, right? Then we form an enzyme product complex, sucrase, glucose, fructose, because we're at the point of breaking that bond. So we have the enzyme still attached, but now we've taken that sucrose and started to break it into the glucose and fructose that form it. And then finally, we release the products, this glucose and fructose, from the sucrase, and now we have sucrase free to go on and pick up another sucrose molecule and do the whole dance over again. So here we see that whole dance graphically. Instead of looking at the words, we can look at the cyclic nature of it. This is why these enzyme catalyzed reactions are so productive, because that enzyme can work on many substrate molecules as it turns them over and allows for a new substrate to bind the active site. So we have our active site. If we start at the roughly 9 o'clock clock position, then our active site is there. The sucrase is ready to form that enzyme substrate complex by accepting a sucrose substrate into the active site. We add the water, which is necessary for the hydrolysis of that bond. So now we've reached the point where we have the enzyme product complex because we've added the water and thereby hydrolyzed that glycosidic linkage. We have the enzyme still holding on to our products now, the glucose and the fructose, and then we allow the products to go free, and that frees up the active site of the enzyme to enter the cycle all over again. So if you didn't make so much sense in words in the previous slide, hopefully with the picture it makes a little more sense in this slide. So we have a couple of different models for enzyme action. The first, and probably the one that you learned about way back in middle school and then again in high school, The lock and key model is the more popular model that's taught, and that's a rigid substrate binding to a rigid enzyme, much like a key fitting into a lock, and that's what we showed in the previous slide there with our sucrase. The active site was ready to accept the sucrose substrate, and then when it released the glucose and fructose, it had that same shape ready to accept another sucrose molecule. That's, again, for the highly specific type of reactions that we talked about, that's a common type of enzyme action. We can also think about the induced fit model, and that's a more dynamic model of enzyme action. In this case, the active site's flexible enough to adapt to the shape of the substrate, so clearly this might work a little better with the broader, either the group or the linkage specific enzymes, because here you have an active site that is not fixed for a particular substrate, but it could be adaptable to maybe a few different substrates that are similar, or even a whole class of molecule that has a particular linkage type. So induced fit model has the substrate and enzyme working together to acquire that arrangement that lowers the activation energy, rather than already have the enzyme pre-fabbed ready to accept the substrate in its current specific form. So there's the big difference. Lock and key model, it's ready to act, and it's only ready to act on that particular substrate, whereas the induced fit model, once that substrate approaches the enzyme active site, there's some modification to that site in order to accommodate that particular substrate or that group of substrates that can be induced to fit that enzyme active site. And just like we did before for the disaccharide hydrolysis, here we see a nice cartoon for induced fit, right? The enzyme substrate complex for disaccharide hydrolysis showed the sucrase and the sucrose, and that was in line with the lock and key model. Here we see an active site. If we look at roughly 12 o'clock, the enzyme has sort of the bottom of a hexagon active site. The substrate is spherical, or at least circular, since we're in 2D here. And we see as the substrate and enzyme start to get to know each other, the substrate starts to move toward that active site. Notice the active site and the substrate both change in shape. So we have conformational changes occurring at both the enzyme level and the substrate level to form that enzyme substrate complex. And then we form now the enzyme product complex, release the products in that next step, and have that enzyme go back to its original active site that doesn't look like the right fit for the particular substrate, but as we saw before, they'll come together in such a way that both change enough to have a nice tight fit. Okay, so before we end this first lecture here in Chapter 20, let's end with a learning check, as we so often do. For A, you're asked which is the active site, and then you have a few options there. And then for B, in the induced fit model, what is the shape of the enzyme when substrate binds? And then you have a couple choices there, so hopefully both of these are fairly simple, but go ahead, pause the video here if you need to think about it. And when you're ready to check your work, please start us back up. Good luck. Okay, so hopefully A was pretty straightforward. The active site is choice 2, a section of the enzyme. It's not the entire enzyme. It's certainly not the substrate. The active site's concerned with the enzyme, and it's a particular section, often a very small section of the overall enzyme. For B, the induced fit model, when we have that substrate bind, the enzyme active site adapts to the shape of the substrate, which also changes, as you recall from our earlier discussion. So lock and key model, everything would stay the same, but in the induced fit model, we have the enzyme adapt to the shape of the substrate, which also undergoes a conformational change. So hopefully those were two easy ones. They were both the second choice, so hopefully that didn't throw you off. But if you are struggling, please do reach out. This is unfortunately as easy as it gets. So hopefully it's going well, and we'll see each other in the next lecture. But if you're struggling here, we should probably get in touch before we move on.